Good morning, good afternoon, and good evening, everyone, and welcome to the Scientist.com webinar titled Applications of Single-Cell Sequencing, Innovative and Tailor-Made Services. I'm Andrew Davies from the events team here at Scientist.com, and I'll be your host today. This webinar has been sponsored by Single Cell Discoveries, so a big thank you to them for helping to make this event possible. We're very fortunate to have Bastian Bale and Dylan Moyman from Single Cell Discoveries with us today, and they'll provide an introduction to single-cell sequencing and its potential application to different research questions and include some user cases. So with that, I'd like to welcome our first presenter, Bastian Bale. Thank you very much for joining us today, Bastian, and the floor is yours whenever you're ready.
Thank you very much, Andrew, for this kind introduction. Yes, my name is Bastian Bale, and I'm the Head of Business Development at Single Cell Discoveries. And I'll be taking you through the first half of the presentation, after which I will introduce my colleague, Dylan, who will take who will present the second and last part of the presentation. There we go. So applications of single-cell sequencing and our innovative and tailor-made services. Who will be presenting today? So my colleague Dylan Moyman, he is the head of the R&D department at our company. He will be presenting the second half of the presentation. But I will start. My name is Bastian Bell. I'm the head of business development, and I will start with the first half of the presentation. I'll tell you a bit more about our company and about our services that we provide, and then Dylan will take over. So who are we? We are a dedicated single-cell sequencing CRO or contract research organization. We were founded in 2018 as a spin-off from an academic core facility based in Utrecht in the Netherlands, and that's where we are still based. And as you can see on the right-hand side, we currently work with a passionate team of 18 scientists, and as of now, we are actively recruiting more people to join the team, so we are growing rapidly. And it's our mission to make single-cell sequencing technologies available to everyone, everywhere. We do that for a number of clients. We work through Scientist.com, through the Scientist.com platform for Roche, for AstraZeneca, for Merck. We also have clients next to the Scientist.com platform that we're very proud of having, among which are Genmab and AstraZeneca and GSK. And apart from that, we also work for a number of universities across Europe and the rest of the world. We currently have more than 250 happy customers in more than 20 countries. Most of our clients are based in Europe, as our labs and offices are based in the center of the Netherlands. We have a few clients in China and a few in the US, and we hope to grow those in the coming months and years. We serve clients at over 100 institutes and companies, and we have a nice mix of academic, clinical, and commercial clients. So I'm happy with the results of the poll that Andrew just showed, because as we saw, most of you do not have experience with single-cell sequencing. So I'd like to touch upon what single-cell sequencing actually is before I describe what services we offer. So single-cell sequencing, a common analogy that is often used is the smoothie analogy. And if you compare single-cell sequencing with bulk RNA sequencing, so more traditional RNA sequencing, then this analogy works very well. So let's imagine that somebody gives you a smoothie. You have a blindfold on, so you cannot actually see what color the smoothie has. And then the person lets you taste the smoothie and asks you if you can figure out what ingredients were used to make the smoothie. Especially if you're not able to see the color of the smoothie, it will be very difficult to exactly pinpoint what the individual ingredients were that were used to create the smoothie. You might be able to figure out that it contains a bit of orange juice and maybe a little bit of strawberries, but determining the exact composition of the smoothie, let alone how much of each ingredient was used to create the smoothie, will be very, very difficult. And this is exactly what happens when you do bulk RNA sequencing. When you do bulk RNA sequencing, you typically analyze the mRNA or the RNA contents of tens of thousands to millions of cells at the same time. So what you get is an average transcriptome profile of all the cells combined. And it doesn't give you any information at the level of single cells, individual cells. And that is where single cell sequencing comes in. So going back to the smoothie, if you were to apply single cell sequencing to the smoothie, you will actually be able to determine which ingredients were used to generate the smoothie, to create the smoothie, and also in what approximate ratio they were used to make it. The same is, so if you apply that to your sample, you get a transcriptome readout at the level of individual cells. So your resolution of the data that you generate increases significantly compared to a more traditional bulk RNA sequencing experiment. Single cell sequencing is a rapidly growing field, as you can see here. So this was updated yesterday with fresh numbers from PubMed. These are all the publications with a mention of single cell sequencing in either the title or the abstract, as downloaded from PubMed. And as you can see, the number of papers since the original publication in 2009 about single cell sequencing has increased, has doubled roughly every year, and is also growing rapidly this year. So how does single cell sequencing work? And there's no matter what technology you choose, there's a generic 5-step process that that can be applied to any single cell sequencing technology, which I would like to highlight here. So the first step is to generate a single cell suspension. So if you work, for example, with tissue or tumor tissue or tissue material that you would like to analyze, then the first step in the protocol is to separate the cells from each other, to make a suspension from single cells or single nuclei, as we will touch upon later, because that's the first step that you need to do before sending us the sample. The second step in the process is to isolate or separate the cells from each other, to prepare them for the third step, which is to barcode the cells individually. So barcode the genetic material from each cells individually with a cell-specific barcode before you amplify it. And this second step, isolating the cells, can be done in different ways. And I'll touch upon that later in this presentation. The fifth step is the library preparation and the sequencing of the material. And the final step is the analysis of the data. So this is a generic 5-step process for any single cell sequencing platform or technology. So how do we help with our single cell sequencing services? Well, let's first define the problem. So the problem with single cell sequencing is that there's multiple technologies to choose from. So which technology should you actually choose? If you want to do it yourself, if you were to do it yourself, you need to invest in expensive equipment, but more importantly, in people. So you actually need to train people to do it. It's not your standard. It's not your standard life sciences assay. It's quite complex. So you need large investments in both people and equipment to be able to do it yourself. And the analysis of the data is highly complex. So how do we help? By providing an end-to-end service for single cell sequencing. So we invite you, if you have a project that you would like to discuss, or if you are interested in outsourcing your single cell sequencing services to us, we invite you to schedule a free consultation with us. We will then discuss your biological question, the design of the experiment. We will discuss your budget, and we'll be able to advise you on the best platform or technology to choose. The next step, we will process your samples in our wet lab in Utrecht in the Netherlands. And finally, we help you with the data analysis. So a preliminary analysis of the data is always included in our services. And as I will touch upon later, we will also be able to help you with additional analysis if necessary. So our technologies, we offer different technologies, not only for single cell, and I'll touch upon them briefly. So the first technology we offer is called SWORD-seq, which is a cost-effective plate-based platform for single-cell transcriptomics, and I'll highlight a bit more about how it works in a subsequent slide. Next to SwordSeq, we offer VASA-seq, is also based on plates, but then for full length and total RNA single cell sequencing. So it does not only capture the 3' end of the mRNA, it captures the full length of the mRNA molecule and also captures non-coding RNA species. And this is an exclusive technology that we have licensed from the Hubrecht Institute to offer as a service. Of course, we offer 10x Genomics single cell sequencing services, and we are very proud to have been the first commercial certified service provider in Europe for 2 of the 10x Genomics technologies. We offer highly cost-effective bulk RNA sequencing services, and later this year we will also start offering 10x Genomics Visium for spatial transcriptomics services. So let me dive into a bit more into the details of some of the technologies that we offer. So SwordSeq, as I said, is a plate-based single-cell sequencing methodology. It's based on these 384-well micro-well plates that you see on the right-hand side. FAC sorting is required. So what we do is we prepare the plates in our lab with barcoded primers. Each well contains primers with a well-specific or cell-specific molecular barcode. We ship the plates to our client, and they use their own FAC sorting facility to sort a single live cell into each of the wells. After that, they send it back to us, and then we generate a single-cell sequencing library out of it. SwordSeq is suitable for low cell numbers, or if you need to do a specific sort, or if you're only interested in a very specific cell type, then SwordSeq might be a very good option because you can sort directly into the plates instead of in a tube. And it's flexible and modular. So we advise clients to sort at least 2 plates per condition or per sample, but preferably even more, because that allows you to increase the number of cells in your data set at a later stage if necessary. 10x Genomics is the next technology I'd like to highlight. So 10x Genomics offers high throughput single cell transcriptomics, high throughput single cell sequencing. 10x Genomics are the market leader. They're a company based in California in the US, and they are the market leader in the single cell field. So most of the papers that are published today with single cell data have been the data has been generated on the 10x Genomics platform. We offer 2 of their technologies currently: the 3' single cell gene expression solution and the 5' single cell immune profiling solution, which allows you to map the TCR and or B cell receptor, the T cell receptor and or B cell receptor repertoire of your samples. And as I mentioned before, we are very proud to have been the first commercial certified service provider for both the 3' and the 5' solutions from 10x Genomics in Europe. Last, I would like to highlight is our bulk RNA sequencing technology. As I said, it's highly cost effective, so we don't use any commercially available kits. So we can offer quite cost effective pricing for our bulk RNA sequencing services. And we can start with as little as 100 cells per sample for the same price as for more cells. So no low input pricing differences for this. So let me touch upon the data analysis, because that's quite an important part of the whole single cell workflow. And before I dive into what we actually deliver, let's talk very briefly about what it is that we actually generate. So how does the data look when we generate it? Well, essentially this is all that we generate. So when we process the data, we map the data, we generate these tables, and this is called a count table, where each row represents a gene. So there's 20,000 rows in this case, and each column represents a cell. And in this experiment, 4,000 cells were used. So this is a very large table with values, and these values represent the relative expression of this particular gene in this particular cell. So even though this is quite a simple table, this is not very effective for us humans to interpret this. So therefore, there's there's some advanced pipelines, some advanced algorithms to make sense of this multidimensional data that results in something like this. So this is an example from a paper that was published in 2020 by the Clevers Group from the Hubrecht in the Netherlands. And what they did is they used our services to generate single-cell sequencing data from snake venom gland organoids. So they made organoids from actual snake venom glands. And they used the single-cell sequencing technology. They used SORT-seq to determine the cell types that were present in the organoids and to compare the cell types present in the organoids with the cell types present in the original snake venom gland. And the result is it looks like this. So this is what is called a T/S/E plot, and what it basically does is it tries to generate an image based on the table that you saw on the previous slide. Each dot represents a single cell, and the cells are clustered based on the relative expression of all the genes that you can find in the table. And then what you can do is you can start to look at the clusters that are defined by the algorithm, and then try to make sense of those. So you need to look at what genes are differentially expressed in a certain cluster compared to the rest. And to give you an example is, so on the left-hand side, you can see the individual clusters. On the right-hand side, you see an expression profile of a single gene overlaid on the same T/S/E plot. And this PRSNTX gene is a gene encoding for a toxin. And you can see that the expression of this gene is highest in the bottom two clusters. And therefore, the researchers were actually able to figure out that the bottom two clusters are actually toxin-producing cells. And this is what you can do with all the genes. So you can look at the relative expression of a gene on top of the T/S/E plot that is generated by the organism. So this is very brief what the data looks like that we provide you with. So what do we provide you with? If you choose to work with us for single-cell sequencing, you will receive the FASTQ data, the raw sequencing data, of course. You will receive QC reports, and we will provide you with a preliminary cluster analysis and a preliminary differential gene expression analysis based on your data. We will also provide you with a free BIOTURING license. So we have two options. If you work in academia, you can get a limited license for free with an unlimited time amount. What we will do is we will prepare the data in Surat, which is a standard single-cell sequencing analysis pipeline. We will prepare the Surat object on our BIOTURING server, and you can easily log in with your account and with our license and download the data. It will be ready for you, download it, and then you can visually explore it using the single-cell browser from BIOTURING. For commercial clients, we can offer a non-limited functionality license, but with a time limit of 4 weeks. And if you want more options, more functionality as an academic customer, you'll need to contact BIOTURING, or if you need more time as a commercial client, you'll need to contact BIOTURING as well. But we're very proud because BIOTURING is really top of state of the art when it comes to visually interacting with your single cell data. Like I said, we can also help you with custom data analysis. So a few examples of what we can do are we can recluster, we can do cluster analysis and cluster characterization for you. We can do differential gene expression analysis, gene set enrichment, trajectory inference and pseudotime analysis, or integrate new data sets that we generate with publicly available data sets or other data types if necessary. And this is just an example of what we can do. We have a team of very capable bioinformaticians that can help you with your data analysis needs. So to summarize, why would you actually want to work with us? Well, as I mentioned in the beginning, we are a dedicated CRO for single-cell sequencing. So we do one thing very well, and that is single-cell sequencing. We have an expert team, so we have many PhD level team members, and Many people in the team have multiple years of experience in single-cell sequencing. Actually, one of our founders started doing his first single-cell experiments in 2012 already, which is quite early. We offer cost-effective services. We are small, and that means we're agile, so we can help you with tailor-made projects. So we can team up with other biotech vendors, for example, to help you with cell line stimulation, compound stimulation, and then work with those cells that are generated by one of our partners. There's multiple technologies that we can offer you, so plate-based SWORD-seq and VASA-seq or 10x genomics, high throughput, and we offer extensive bioinformatics support to our clients if necessary. We always advise to start small. So single-cell sequencing is quite an expensive technology, and it does make sense to start with a small-scale pilot project or small-scale proof of concept. And scientist.com is the ultimate platform to get started quickly with that. So this proof of concept or pilot project can act as a technical validation as well as a scientific validation. So does it even make sense to use single-cell sequencing? Will it answer my biological question if I do single-cell sequencing? So based on the data that we generate in a proof of concept or pilot, we can decide, or you can decide, how to proceed and if you want to scale up. So how to get started? So the first step is to schedule a free intake meeting with us. You can actually click. This is a clickable link. You can directly book a consultation with us in our calendars to discuss your project details, sample types. Then we'll advise you on the technology to choose, and eventually we can also discuss your budget, and we'll be able to help you with the budget. And those are my slides. I would like to give the floor to Dylan, because he will speak about what's next in our R&D portfolio and about the applications of single-cell sequencing.
Yes, thanks, Bastiaan, and thanks all the attendees for coming to this webinar. I would like to highlight some of the methods that we are currently working on and might be of interest to the audience. So as Bastiaan already in a couple of slides before mentioned, that even though our name is single-cell discoveries, we actually do quite a lot of other sequencing methodologies as well, including bulk sequencing. And one area that we're quite interested in exploring more is improving the current bulk sequencing methods that are out there. So we try to use our expertise in single-cell sequencing and all the molecular tricks that we have sort of learned along the way to not just apply them to single-cell sequencing, but also use them to bulk sequencing and improve the throughput and cost of lower the cost of bulk sequencing. So one of the ways to do this is by using the robotics that we have from our single-cell sequencing pipelines, and use the extremely low volume preparation steps that are used for single-cell sequencing. This benefits both the quality and also the cost of the reagents will also go down. This will allow us to process hundreds of samples at a time using these 384 well plates, compared to the commercial kits that often are limited to a handful or a tens of 20 of samples you can process per kit. And indeed, the applications that are ideally suited for high throughput bulk sequencing are, for instance, CRISPR screens that are done sometimes in the genome or transcriptome-wide. CRISPR screens are being done where one can read out the phenotypes using microscopy or a fluorescent reporter. However, many more subtle phenotypes are lost in that sense. And sequencing the actual transcriptome to get a more more sensitive phenotype is often way more suited for those types of experiments. And a similar thing is true for compound screens. And in addition, we are noticing more and more customers are interested in taking large patient cohorts with certain diseases and having matched healthy participants in there as well, and trying to screen and sequence a large proportion of these cohorts to try and find any molecular differences between them. So having a high throughput and low cost, low volume solution for this is actually ideal. So this is something that we are currently working on hard to get that to the next stage. So another thing that we are working on is automated nuclei isolation. And this is something that is quite interesting because many customers do not want to dissociate or do not have the expertise to dissociate their own tissue. So this will allow for a standardized way to isolate not single cells, but nuclei that are then immediately usable for single cell sequencing. So we can then process or perform single cell nuclei isolation from fresh frozen tissue. That is quite important. And we can isolate these nuclei in a very, very short amount of time, like 7 to 12 minutes. This is done mechanically rather than using many, many enzymatic steps. So this is also done cold. This is not a room temperature or incubation at a higher temperature. This is all done at very low temperatures. And there are many, many applications for this. So there are certain tissues that are simply unsuitable for single-cell RNA sequencing. It is not possible to do a proper single-cell dissociation because either many cell types are lost along the way, or the dissociation takes so long that the quality of the RNA and the cells can no longer be guaranteed. So for those types of tissue, brain is an obvious example of this, but there are many, many more. Nuclear isolation is definitely the best option. There are also samples that require immediate storage because they are highly, highly perishable. So for those tissue or samples, they can simply be snap frozen, immediately sent to us, and we will then isolate the nuclei and sequence those. And lastly, what I already hinted on, it is non-trivial to do a proper single-cell dissociation experiment for many organs and tissues. Simply, they're quite tough. There's a lot of different ways, a lot of different enzymes, a lot of different things to optimize. Not everyone has the time, manpower, and expertise to do this. So this is a very streamlined and easy solution where tissue and cells can simply be frozen, sent to us, and we can actually process everything from there. And the last technology that we're working on that I would like to highlight is more relevant to people interested in immune biology. We are working on doing plate-based TCR and BCR sequencing. So this is what it means is indeed single-cell sequencing of VDJ regions, and we use here FACS-sorted 3-letter well plates as an alternative to the 10x Genomics platform. Because one of the issues is that to detect these rearrangements in very low cell numbers, it is very hard to, often not possible currently, because the 10x Genomics immune profiling solutions, they require often more than 100,000 cells to actually run. And there are many applications where you simply you never have access to that many cells. For instance, there are very complex T-cell, B-cell FACS experiments. We have had clients who do very high dimensional sorts where in the end you end up with such a low fraction of cells you do not have enough to load on a 10x chip. A lot of dextamer sorted, so antigen specific profiling. Also, you often end up with a couple of hundred cells, and that is simply not enough for 10x Genomics profiling. Even if you do have enough cells, it would still be advantageous to go for a plate-based method compared to a microfluidics-based method, because we have noticed that for almost all of our plate-based methods that we obtain a higher sensitivity compared to microfluidic methods. So we often detect at least twice the amount of unique genes compared to microfluidic methods. So there are applications where someone needs a higher sensitivity. So plate-based is actually a preferred choice. So I've just given you an overview of some of the tools and methods that we're working on, but I'd like to now talk briefly about some of the applications of single-cell sequencing, practical applications, things we've collaborated and worked on. Because what are some of the ways you would use single-cell sequencing? Bastiaan has given a very good introduction about sort of the fundamental principles of single-cell sequencing, how do all the different methods really work. But why would you choose single-cell sequencing in addition to other methods that are out there? So one of the ways to think about it is it's a very high resolution way of looking at gene expression. So instead of looking indeed at an averaged profile of gene expression, you can actually understand much more of the individual pieces that are going on. So one of the main topics that we generally use single-cell sequencing for in collaboration with our clients is people who have very heterogeneous manipulations done to their cells. So for instance, what I've talked about before, drug screening or CRISPR screening, generally these things are not done in a way where every single cell will have the same response to the drug, or even you generally don't have a single guide RNA for your CRISPR screens. You often have many, many different guide RNAs, and these can be done in one single experiment. And single-cell sequencing can be used to read out the different phenotypes that are corresponding to those. The similar thing with viral infections is something I will talk about later. There are many different ways of inducing certain phenotypes in cells using viruses, but viruses do not infect every cell equally. Certain cells will simply take up more of the viruses, some of them will not. And it's very hard to correlate phenotypes if you don't know what your viral load is on a per cell basis. And even without actively manipulating cells, if you grow cells in a cell culture plate or in any other way, cells generally are not all the same within that cell culture media or within their position within the plate. Cells that are on the edge of the plate are in a very different environment than cells that are in the center of the plate, or even cell density can have a large effect. So single-cell sequencing can help you to understand what type of different responses there are for cells in these different environments. So that is one aspect. The other aspect is that there are many poorly characterized cell systems. And the two main ones actually that we have experience with is novel model systems that often academic clients have developed, and they would like to have a more unbiased understanding of which cells are in there and what would those cells potentially be doing. And for more therapeutical applicable models such as whole organs, that's also something I will talk about in the next couple of slides. Even in human organs, it is not well understood what different cell types there are and what they might do. So using a high throughput single-cell sequencing platform such as 10x is an ideal way to get an unbiased view of all the different cell types you have in your system. So I will now talk about a couple of different examples that we've worked on. These different case studies. So the first one is a client who wanted to develop a novel drug to treat a liver disease and hepatocellular carcinomas. So there still are currently very few drugs out there that treat advanced liver diseases. So this client wanted to use single-cell sequencing to get a little bit of a better understanding about what types of drugs could be used when certain types of damage has been done to the liver. So indeed, they use single-cell sequencing to figure out drug responses in a heterogeneous Hepatitis-C infected cell culture model. So they took a cell culture model that mimics the sort of in vivo liver situation. They used hepatitis as a way to induce damage in these liver cells, and then they did a drug screen to see which drugs could counteract some of the damage that was done by hepatitis-C. So this is indeed a very good example of why single-cell sequencing is essential to be able to correlate the drug response and the response of the cells to the hepatitis infection. So I hope you can sort of appreciate or see this picture that is here, the table that's here in the middle. If you have a couple of things going on here. So you have the white squares that are on the left side, and those are representative of cells that were not infected by hepatitis-C. Even though indeed they were exposed to it, but just by chance they did not take up the virus. And on the right, you see black bars. Those are cells that did take up the virus, and they took it up also in a heterogeneous manner. So some cells have accepted more of this virus compared to others. So the blue histogram that are on top of that represents the viral load, which all the way to the right, you will have the highest viral load, and it sort of tails off towards the middle. And what they've done is they've done single cell sequencing for a handful, about 40 cells per drug treatment, and the drug treatments are on the right. There's a handful of different drugs that were screened, and what this would then allow you to do is figure out what is the response for these cells when they have a high viral load and they're treated with a drug compared to cells that have a low viral load, for instance, and were treated with the drug. So it allows for this very highly accurate correlation between the disease-causing treatment and the drug treatment that is supposed to counteract this. So in the end, the client came to us in the end to use a different technology, because originally they had used SmartSeq to be able to generate this data. But SmartSeq, however, is a relatively low throughput method. They have been able to sequence about 480 cells. However, they wanted to reproduce all their results in a patient or in multiple patients. And they ran into a problem where they could not get enough numbers to actually get accurate information from there. So they came to us to use our SortSeq technology, which is cheaper and more high throughput than SmartSeq. So in this study, they went up to about 2,300 cells using SortSeq. And this actually allowed them to generate these TSNE or UMAP plots. So you can see here on the right, and which then also allowed them a higher resolution and figure out where these gene expression changes that were caused by the drugs were actually occurring, and they figure out that they were actually occurring in the macrophages. So that really gave them much more insight about how this treatment was actually working. So the next case study that I'd like to highlight is a group in Holland, I think in Limburg. They wanted to understand the human cornea, and because there was at that time a very limited understanding about the amount of cell types are actually there, and they wanted to understand what potential therapies could be generated for different diseases that were causing blindness. And because they didn't understand what type of cell types were present, they could also not develop therapies targeting these specific cell types. So this is a good example of a case where a sort of more high throughput approach using 10x genomics is more applicable, because this allows for a more unbiased mapping of these different cell types. And one of the main things that we did different with them compared to a lot of our other clients is that what they did is they Methanol fixed these cells after they dissociated them. So in this overview, you can see they took human cornea from donors, they dissected the tissues, they then dissociated these tissues into single cells, and then they methanol fixed them. So that allows, one, for the cells to be stored for a long period of time, but it also fixes the cells in their current state, so there are less transcriptomic differences that can occur, and also any cells that are more fragile that you might lose over time are also then preserved. So it maximizes the possibility of capturing all the different cells and cell states that you have. So these cells were then shipped to us. We rehydrated these methanol fixed cells, loaded on a 10x platform, and then sequenced it and generated the data for them to look at. And in the end, what resulted from this data was quite interesting, is that on the left you see a UMAP or TSNE. Basically, it's a similar thing, a plot where you have three large clusters representing three cornea layers. However, on the right you have a picture showing that the cornea actually consists of 5 different layers. However, it turned out indeed 2 of these layers are non-cellularized. So in the end, we captured all the cellular layers that were present in the cornea. And in this study, they also in the end found a specific stem cell niche that was present, or at least was visible in these single cell sequencing data, which then gave them a handle for any therapeutic drugs that they wanted to explore afterwards. So these were the 2 different studies that I wanted to highlight. And I think we can open the floor for questions now. Thank you.
This marks the end of our presentation. So if there's any questions, please feel free to use the Ask a Questions pane that you can see on your screen. I would like to highlight a few options for you to get in touch with us if you're interested in working with us. So you can send me an email by clicking on the link here. You can go to our website, or you can book a meeting with us directly into our calendar by clicking on the link that you can see on the screen.
A good place to start here is maybe, how do I know if I need single cell RNA sequencing or regular bulk sequencing?
Yeah, that's a very good question, and it's as with many questions that we get from clients or potential clients, that depends on your biological question. So if you need, if there's a reason for you to need the data at the resolution of single cells, then of course you need single cell sequencing. But sometimes the biological question doesn't need single cell resolution. So then bulk RNA sequencing is definitely a very good option, because it's a factor 10 cheaper than single cell sequencing. So it really depends on the biological question. So if this is something you want to discuss, please feel free to schedule a meeting with us, and then we can discuss your project in more detail, and we can advise you on the best approach.
Great, thanks very much for that. Maybe this is a good one, might address questions a number of people have. What kind of tissues and species can you process with single nuclei sequencing? Are there any limitations to those?
Yeah, single nuclei sequencing. Dylan, maybe you can answer that. What?
For single nuclei sequencing, so in principle, many more tissues can be used for single nuclei sequencing compared to normal, let's say, single cell sequencing, because you are skipping that dissociation step. But there are still some tissues that have a high fat content, high lipid content, that are quite hard. So skin would be one where we know that is still a big problem. But in general, it is easier to do than the normal standard single cell sequencing. So tissues that would work well are brain, for instance, like let's say a lung, even pancreas, I think is relatively doable. So it's more the other way around, I guess, that there are some exceptions for single nuclei sequencing, while for normal single cell dissociation, you have to take a lot more care.
Okay, great. This one, sort of related. The person says, sorry they joined late, so they apologize if they've already covered the question. I don't think they have. Their question is, have you tested single cell sequencing on fetal heart specifically? And if so, can it be done on fixed cells?
Yeah, I can take it. I don't know if we've done fetal
I
heart.
don't think.
And
I don't think so.
Sorry. Okay. Yeah, that's also but my question would also be fetal heart. I guess from which organism? That will be so perhaps, one thing. But in addition, sorry.
Yeah, perhaps they can follow up with some more specific details and get in touch with you through these. You can click on these links here if you want to contact Bastian or the single cell team, and they can follow up for a specific application. Yeah.
One general comment on heart tissue, cardiac tissue, is that typically cardiac cells are quite so not fetal cardiac cells, but in general cardiac cells are quite large, so they typically do not fit through the tubes, tiny tubes of the 10x genomic system. So we've done quite a number of single cell sequencing experiments with the SORT-seq platform. So sorting cardiomyocytes into the plates, and that has yielded very good results. So if you're interested in discussing this, please feel free to reach out to us, and then we can discuss your project in more detail.
We missed one part indeed. So you asked, can it be done on fixed cells? And like I mentioned before, we have experience with methanol fixation. So that will likely work, but we should probably talk about this if you're interested in it. Sorry, Andrew.
Oh, no problem. I was actually just going to lead into that next. Another question from someone else here relates to this to some extent. So do you work with FFE blocks? I presume that's formalin fixed, paraffin embedded blocks.
We cannot do single cell sequencing on FFPE block material because you cannot generate a single cell or single nuclei suspension from FFPE blocks anymore. But for Visium, we will eventually be able to work with FFPE blocks. So I very briefly mentioned Visium, which is a spatial transcriptomics technology from 10x Genomics. They do have an FFPE version of the protocol that works on human and mouse samples. And when we launch the Visium spatial transcriptomics service later this year, we will also be able to work with FFPE blocks.
Okay, wonderful. Just a note, another question on that's somewhat related to that. I believe Dylan mentioned a number of other services that you're working on. Is there a particular timeline for when those are available? And someone is asking if it's possible to send you test samples to try out some of these methods.
Yeah, so the single nuclei isolation service, we hope to be able to offer that in a few months from now. The other two methods, so the screen-seq, the plate-based bulk RNA sequencing, which we aim to price quite significantly lower than a regular bulk RNA sequencing service. And so the screen-seq and the plate-based VDJ, we hope that we will be able to offer that this year or early next year. But the first additional R&D project that will be transferred to the services team, so from Dylan's team to the services team, will actually be the single nuclei isolation service that we will offer as an extension to our current 10x Genomics services. So that means that clients will be able to send us fresh frozen material, and that we will take it from there.
Okay, great. So watch this space. Question here: how well could you detect I believe this is long non-coding RNAs?
Yeah, that's a question I can take. Well, I guess it depends on if they're polyadenylated, we can basically detect them with all the standard methods. So that is not whether it's 10x or short-seq. If you're interested in non-polyadenylated non-coding RNAs, then VASA-seq is pretty much the only option, because that indeed detects everything, whether it's polyadenylated or not. But as long as they're indeed long enough, so we're not talking small RNAs, but anything above 200 nucleotides is detected just as well as every other mRNA that is out there.
All right, great. Another question here: can you please describe the difference regarding what we can achieve from single cell sequencing and spatial sequencing? So I guess the question is, what different information are those two methods providing?
Yeah, I can take that question as well. So I do have to put a little asterisk in there. This is what is currently possible, and this is currently what the difference is. So with single cell sequencing, you no longer have information about, of course, which cell was next to which other cell. So you no longer know any potential interactions between the cells. However, you do get a much higher sensitivity and higher resolution from the gene expression from each of these single cells. With spatial sequencing, you then know the actual tissue context of the cells that you're interested in. So you know if a certain immune cell is next to a cancer cell and it has a certain expression profile, you can then use that information potentially for therapeutic or diagnostic possibilities. However, currently the resolution of spatial sequencing isn't at the single cell level yet. So if you really want, it depends on what your preference is. If you really need single cell information, I would not do any spatial sequencing. However, if the context of the tissue is really important to you, more so than the single cell information, then spatial sequencing is the way to go nowadays.
All right, great. Maybe in the interest of time, we'll make this the final question. And I'm not sure if you can address this globally. It may depend on the project, but what is the cost for testing each sample, such as a tissue sample, for example?
Yes, I'll address that. The cost really depends on the exact setup of your experiment and what technology you choose. So please feel free to reach out to us directly, and we'll be able to help you with a project-specific pricing overview. based on the number of samples and based on the technology that we advise you to use, so either SWORD-seq or VASA-seq or 10x Genomics. So we can discuss this in person.
All right, wonderful. Before we go, I'd just like to thank you both, Bastian and Dylan, for the wonderful presentations and sharing all your insights through the Q&A here today. It's been a pleasure having you with us.
No, thank
Likewise.
you, Andrew.
Thank you very
Thanks
much,
to the participants.
Andrew.
Wonderful. Thank you both. So in closing, we hope that you've enjoyed this scientist.com webinar, and we'll hope to see you again next time. Thank you very much.